Good evening, class. It's an opportunity to welcome you guys to our online class. Part of it, about 313, animal ecology. For 3 weeks now, we have gone far with animal ecology. We'll talk about historical background of animal ecology, talk about ecology of terrestrial animal and aquatic animals. We'll talk about growth rates and age structure of animal population. Today will be population dynamics, measurement of population dynamic. I've just checked the course materials and realized that some of the students have not been able to do some of the class group discussions with questions that were put up. I don't know what the reason may likely be. All right. Today's class, as we all know, we'll be looking at measurement of population dynamics, the life tables, and the K factors analysis. That's what we shall be considering today in our class. And already I have a PowerPoint that I want to quickly share with you, the student. So I'll be sharing the PowerPoint now. An opportunity to welcome everybody to class bar 304, which is general ecology. Take this opportunity to meet students who are already involved from what we shall be doing this semester. We shall be having lectures already. We've had about 1 or 2 lectures on the study of the ecosystem. We also have plans on the cycling of nutrients in ecosystem. We'll be learning with about 5 other topics that are about the interaction section in ecosystem. We shall also look at population dynamic, which includes statistical that are having population structure. Also put a look at biological communities, different types of biological communities that we have, and students on the different nature communities, the aquatic biomes, and I'm looking at the influence of man. Think that it's a rich biological community. This course in general biology much more to that. All right, that was an intro of introductory video of this course. We'll continue to be having that till we're done with this particular course. So, my name still remains Dr. Senawemeka Kupete, and you can get me on the personal@gmail.com. Today's topic, population dynamics, or rather we say measurement of population dynamics. Like we said, population is just a collection of individuals of the same species that live together in a region. And whereas when we talk about population ecology, what we invariably means is just the study of population and how these population change over time. We have what they call open population and what we call closed. An open population is that type that changes over time. Likely a lot of factors could be responsible for it. And for the closed ones, are the ones that have not been able to change over time. The population can decline because it lacks lots of resources. And, most of these resources can either be biological factors that can affect the increase and decrease of the numbers of species that are found in the population. So the maximum population number or abundance that gets to a level where the resources can sustain is what we know as carrying capacity. When we look at the next chart, we've seen a population that is expanding, a population that is stagnant, and a population that is decreasing. The A, B, and C. It's just an explanation to the other chart that was shown in terms of post-reproductive, reproductive, and pre-reproductive situation, where the pre-productive population is high. Invariably means that such population is expanding. Whereas for B, such population is stagnant because we have a stagnant number of pre-reproductive and reproductive age. Whereas for the last C, population that is diminishing is a population where you have more of post-reproductive, because those have exhausted their reproductive mechanism. So in such population, there is no way it will be expanding. Before we talk about survivorship curve, which is part of population measurement, we have to understand that there are different methods to describe population, different methods that are used to describe population, from field measurement. Could be life table, could be transition matrix, it could be sensor of individuals in a group. These are different dynamics that are used to relate demographic processes to predict the fate of the population. There are a lot of factors that needs to be considered when such assessments are done. It could be conveniences of data or data collection. One, it could be basic assumptions. Two, it could be the way the drive population growth rates. You have to consider this because this will go on to influence whatever method you use in calculation of population or in describing any population. So these three are very, very important. So when we talk about life table, this just describes the survivorship or fecundity of different age, fecundity of different age of animals. When we talk about fecundity has to do with the production of eggs. That production of eggs could be seeds or the young. That's what we talk about fecundity, the ability to produce these things. So when we're using the cohorts, it's very important that this process has to be followed until the last member in this group die. So survivorship curve is used to calculate the relative changes or change in the number of individuals of the cohort between two successive sensors. Used to do what? To calculate the relative changes in the number of individuals of the cohorts between two successive sensors. And this sensor is very simple, since one needs only need to be recognized to differentiate them from one cohort to the other. So, and in this, no marking is needed because every individual is counted, which means that they've survived since the last. So that's just a simple method that we use. So age can be convenient to use in cohort study. And also use age, but does not always play a primary role in development and life cycle of the organism. That people looks at the age and see which of these age fall into the pre-reproductive, the post-reproductive, and the reproductive age. But then, what we're trying to say is that doing such collections of information, it does not affect the development cycle of the organism. Whatever will be as to their life cycles will also or will probably come into fold. So, two kinds of informations are derived from this cohort study. Two, survivorship curves and the population growth rates. That is when we are looking at survivorship curve or survivorship on its own, two informations are derived. Either we're looking at the survivorship curve or the population growth rate. So what do we mean by survivorship curve? Survivorship curve shows how mortality varies with age. Shows how mortality varies with age of the individuals of the cohort. We're looking at age-specific mortality as well as age-specific pincodity. Age-specific mortality, age-specific deaths are dying, and age-specific deaths are reproducing. That is what we all consider in the survivorship curve. So it is due to changes and also capability of the individuals and the variation in the environmental exposure. So we're looking at changes in sustainability, capability of these individuals, and how these changes affect their exposure to the environment. So, like I said, Dever 1947 there described three types of cough. Dever 1947 described three types of cough. And we have the one they call the type 1 coughs. Type 1 coughs occur when survival of young is high and mortality increases drastically towards the end of the lifespan. This is typical of a very protected lifestyle. This occur when survival of young one is high and mortality increases drastically towards the end of the very protected life. In case of 2 survivorship curve, the mortality is constant with age and as in a decay process. But most organisms have the type 3 of curve, of survivorship curve, type 3, where most individuals die when they are young, while other or older individuals are good survivor or survivor. And to portray what we're talking about, that's just a simple chart to show what we're talking about, the three types of survivorship curve. We have the one which are quite peculiar to humans, as we can see. And the one that are constant with age, with birds, and the other one is with frogs. So these are the different survivorship curves that exist in different organisms. Remember that we said that there are two survivorship um, information that we need either for the curve or for population growth rates. The population growth rate is defined as the basic reproductive rate for a generation over its lifetime. And sometimes we use the life table. Life table is best used in study of such reproductive or population growth rate, either in semi-porous animal or monocarpic plants. That's an example of, or sometimes we can also use annual organisms, such as the study of annual plant species, like phyllox, and so on and so forth. They have a simple cohort per year and thus have no overlapping generation. Okay? So either an annual or we're using it for semi-porous animal. It's a simple, I don't have a table yet to show you how a simple life table looks like. It involves series of calculations. So when you check your lecture notes, you will see these calculations on how to estimate survivorship or the growth rate, population growth rate in a population of organisms. So we talk about the static life table. In the case of the static life table, this contains the age group in the population at one particular period of time. So these can be used to calculate population growth only if an assumption is made. In this case, it's used to calculate if an assumption is made. The assumption is that the mortality experience by the cohort at any age stays constant in time. It pays one thing with static life table is the fact that this is used when you now assume that organisms at any age stays constant in time. In other words, the birth rates and age-specific survivorship are assumed to be independent of the actual year in which the observations are made. And this is very rare. It's a very rare assumption to be made, but therefore the conclusion tells us that a cohort should behave, even though if we have observed it and if conditions are constant between years. So so many factors are looked into when you consider a static life table. In population, there is a pattern of growth. Either it's our strategy or K strategy, whether it's an exponential growth or it is a logistic growth pattern. When we talk about our strategy, we're talking about organisms living in unstable environment, hence they undergo rapid reproductions to stabilize themselves. This could be found in so many flies, so many invertebrates in our strategy. Whereas for the K strategy, we're talking about organisms. Okay, living in stable environment, hence they are high in population and do not need to undergo rapid reproduction. Such case could be seen in humans. What are the differences between the K and the R strategy? In terms of habitats, the R strategy, they live in less stable environment. The R strategy lives in less stable environment. Whereas for the K strategy, they live in very stable environment. So what it means that the environment for the R strategy are less stable for them to survive. Whereas for the K, it is very stable. Their conditions are predictable for K, whereas for R, it is non-predictable. Since they reproduce massively, So their lifespans are short for the R strategy. They have a short lifespan. Whereas for the K strategy, they have a long lifespan. Long, can live for years. Whereas for K, at days and sometimes weeks. The sizes, when we talk about their sizes, for the R strategy, it says they are very small in size. Whereas for the K, they are very large in sizes. And already we said they reproduce rapidly, so their reproductive rates are very fast, grow fast, and reproduce fast. Whereas for the K strategy, it reproductive rates are slow, especially for humans, takes 9 months, after fertilization of the eggs for them to give birth to their young one. Offsprings are many for our strategy, and for K, there are very few. But that's of how there could be millions at a time, thousands at a time. Whereas for the K, it could be as very few as you can think of. Their maturity rates are early, very early maturity of the organisms, whereas for the K strategy, they have late maturity of the organisms. Their survival are less for the R, and their survival rates or probability of survival rather are very high for the K strategy. Same with mortality, high for R, and for K, the mortality is low. In terms of predator, whatever relationship, our strategy are subject to predation easily. For the K, they're not subject to predation easily. It's a very few crops that have these relationship together. And for examples of our strategy, insects, bacterials, and so on and so forth are typical example of our strategy. Whereas for K, the humans group, elephants, and other mammals that are known belongs to this group. So now for our class activity, it says which group of organisms exhibit R and K strategy. You do well to, apart from the examples I've just mentioned, you can also give 2 or 3 examples that you know that belongs to this pattern of growth that are found in the environment. All right. Without wasting much time, so already mentioned that there are 2 patterns of growth, exponential growth and logistic growth, and these are the factors that affects population sizes, migration, natality, population size, and mortality, and immigration. And they're the basic. And we'll talk about life table, like we mentioned. Life table is often a simple method of keeping tracks of births, deaths, and reproductive output in a population of interest to us. An example of life tables are shown. We have what they call the stage-dependent life table. We have what they call the statics life table based on living individuals. Statics life table based on mortality record. That's the 3 major examples of life table. And here we talk about population regulation. When population grow, there are things that do regulate this population. Either they are density dependent or density independent factors. And density dependent factors, as we can see, are space, food, water, territory, predators, and prey relationship. These are factors that do affect population. And we'll mention the other one, density independent factors. It could be factors that are not directly dependent on population. It could be temperature, it could be pH, anything we can think of. They are not dependent. It means that they don't directly affect the growth, but they can influence these growth. All right, so class activity 2 is the question: how can population growth of an organism be affected by density independent factor? How can population growth of an organism be affected by density independent factors? That's an assignment class discussion for you. All right. Thank you for the time. I want to stop sharing. All right. Thank you for the time. It's been a great honor to be here tonight so as to share what I've prepared for the class today. I'll be putting up questions for the group discussion. I will likely update, I will update some of the materials that will be needed for this class. So hopefully before Friday, you will get an update on this and what you need to do. Please avail yourself for the course page materials and see how you can also contribute. Thank you. I'm looking forward to seeing you guys next week as we shall be discussing on competition in organisms. Thank you.